Throughout this video we'll walk you through the novel 7Tm phosphorylation assay, which is the first-in-class immunoassay for the quantitative assessment of GPCR phosphorylation that can be performed entirely in multi-well cell culture plates, thus eliminating the need for Western blot analysis. This slide depicts a step-by-step flow chart showing the 7Tm phosphorylation assay that involves immunoprecipitation of affinity-tagged receptors using magnetic beads followed by detection using premium phosphosite-specific 7Tm antibodies as phosphobiosensors. This assay allows quantitative determination of GPCR phosphorylation, particularly when total receptor is determined in parallel using non-phospho 7Tm antibodies, which are included in each kit. The assay can be performed using stably transfected cells expressing the GPCR of interest. Nevertheless, we recommend transient transfection using liposome-based transfection reagents. Cells are then seeded onto polylysine-coated cell culture plates and grown overnight to 95% confluence. Cells are then treated for the desired time with agonist, antagonist, or inhibitor of interest. Cells are lysed in detergent buffer, and lysates are cleared by centrifugation. For parallel detection of phosphorylated and total receptors, the lysate of each sample is divided and transferred to two corresponding wells of U-shaped assay plates. As shown here, a first aliquot of each lysate is transferred to a specific well of a 96-well assay plate that is designated for the detection of phosphorylated receptors. Subsequently, a second aliquot of the same lysate is transferred to a corresponding well of the same assay plate that is designated for detection of total receptors. Antitag magnetic beads are added to each well for receptor immunoprecipitation. Microplates are then washed under magnetic force using a handheld magnetic separation block or automated microplate washer. For detection of phosphorylated and total receptors, premium phosphosite-specific and non-phospho 7Tm antibodies are added to the designated wells of the 96-well assay plate. Plates are washed and enzyme-labeled secondary antibodies are added. After another wash cycle, an enzyme substrate solution is added for detection. The optical density at 405 nanometer is determined using a microplate reader and concentration response curves are calculated. This slide shows a typical result for detection of agonist-driven mu-opioid receptor phosphorylation after stimulation with the full agonist DAMGO. Mu-opioid receptor phosphorylation can be monitored at 4 distinct phosphorylation sites, namely threonine 370, serine 375, threonine 376, and threonine 379. For optimal results, the optical density should be in the range between 0.2 and 1.2. The 7Tm phosphorylation assay can be used for ligand profiling, identification of relevant G protein receptor kinases, and GRK inhibitor screening. Thank you for your interest in the 7Tm phosphorylation assay. For more information, refer to www.7tmantibodies.com or contact us via info@7tmantibodies.com.